Hi everyone. Today I'm going to present to you one of the projects I'm working on where we try to exploit bimodality of gene expression for the enhancement of drug discovery prediction. I will start with a small introduction to the problem and then introduce you to the proposed method, show you some of the results that we have so far, and conclude with some discussion and answers to your questions if you have any. Drug sensitivity prediction is an important problem, and it's a core element towards an efficient personalized medicine process. As you already know, if we have a population of patients, even if it's for the same disease, let's say breast cancer, if we give them the same treatment, all of them, we know that only a small portion of them would respond to that treatment. That's like what we have seen in several studies. And the rest of the population are either going to have like no benefit, or even worse, they might have some severe side effects. So it's important to know which drugs should be given to which patients, and that's the core of personalized medicine. There are many ways to do that. One of the ways is to build models that are specific to each treatment or each drug that can tell us which patients are going to benefit from the drug and which patients are not. And that kind of models can be used across different stages of the drug discovery process. So we could build a sensitivity model for a drug that can tell us whether a cell line is going to be sensitive to a drug or resistant, all the way to, let's say, the pre-clinical trials where we try to know which patients should be included in a trial for a certain drug, because we need to make sure that the patient is most likely going to benefit from the drug and so on. So it's important to find reliable models that can tell us that information. The input to such computational methods there are different types of inputs that we can use, including the mutation data of the patient or the samples, the expression data, and many others. In my talk, I will focus only on the expression data, and it will look like something like this. Sorry, you're going to have the expression levels for different genes across different samples and the drug response to that sample. Okay. So there are many ways to build such models. One of the ways is to make a univariate model where we assume independence between the features. So we try in its simplest form to find correlation between the gene expression level across the different samples and the drug response. And the reason these models are popular is because they are easy to understand. So if we take that simple example and say we looked for a gene that is highly correlated with the drug response in a positive direction, we could say then that as the expression of this gene increases, the sensitivity or the probability that this sample is going to be sensitive to the drug increases, and so on. So it's easy to know what's the factor that is important for that drug. However, the problem is, in reality, it is very hard to find accurate associations or correlations between independent or separate genes and the drug response, because these models doesn't take into consideration any information about the relationship between the genes in the sample. So other groups resort to the multivariate models and the machine learning models, where they try to find a relationship between, as I said, the different genes, the features, in order to associate that with the drug response and have better predictive models. The advantage of such models is that you're going to get better accuracy because you're taking that information into account. However, these models in most cases act as black boxes, so they find the relationship between the genes using their mathematical formulation, which can be different from different types of multivariate models. And it's usually hard to link between those relationships, those mathematical relationships, and the biological aspect of it. So we try to find the best of the two worlds, and in order to find easy to interpret features, we look at the concept of bimodality of genes. that has been used in different applications. For example, it has been used to classify cell lines or samples of breast cancer into ER positive and ER negative based on the biomodality distribution of ESR1, which is the gene coding for the estrogen receptor. So we want to take that concept and apply it to the gene expression data that we have and binarize these expression levels into expressed or versus not expressed across the different samples based on this biomodality distribution. And then relate that to the drug response. So doing that, we build an easy to understand information. We just say that whether like this gene is expressed or not. And by using a complex model that can utilize this information, we can find better predictive models. And we chose to use logical models because those logical models can allow us to find rules, logical rules, that, for example, can say if gene 1 and gene 2 are expressed or gene 3 and gene 4 are not expressed, then the sample is most likely to be sensitive to the drug. So it's easy to understand, it's easy to translate that to a clinical setup. However, at the same time, the logical models are going to try to find relationships between the genes in order to find such rules based on some logical functions. And we chose this method, Logico, that was published in Scientific Report 2016, to be our implementation of one of those logical models. Now we could start by looking at all the bimodal genes and use our logical models to find the rules. However, we thought, okay, we could start with a smaller set of genes that are well characterized and build our models based on those as a proof of concept. So we chose to look at the genes that are related to the epithelial to mesenchymal transition phenotype, which is associated with cancer progression and metastasis, and it's also associated with drug resistance. And interestingly, most of those genes are actually bimodal when we try to look at their distribution in a large set of cell lines, around 900 cell lines from the Cancer Cell Line Encyclopedia. And it's also translated to the tumor samples. This bimodality is still hold there when we did calculate the bimodality over the tumor samples in TCGA. So these are well characterized genes that we can start with, and they are small, so we can know more information about what's happening to them. We decided to do a large-scale analysis based on 3 large pharmacogenomics data where we have drug sensitivity data, CTRBB2, GDC1000, and GCSI data sets. And we analyzed those data sets and mined them using the pharmacogenetics data, which is an in-house package. It's already published in Bioinformatics and publicly available. So to do that, before we build the models, we try to see whether this bimodality classification is going to hold across the different types of data sets. So as an example, we can look at CDH1, which is one of the EMT genes, and look at the green bar, where we try to compare between the data coming from CCLE and the data coming from GDC1000. And we classify all the samples based on the bimodality of CDH1 to be expressed or not. And we do the same thing in GDC1000 and try to find the agreement between the two classifications. So we want to see whether the classification of the bimodality is going to be stable across the data sets. And we found that using the microarray data of GDC1000 and CCLE, we found the most stable agreement between the classifications. However, for the other platforms, we start to find some discrepancies. So we focus on only those data sets, and here I show another metric of the same knowledge. It's the F1 measure. And from those also we chose 8 EMT genes to be our features to our models. So on GDC1000, we built model for each one of the drugs. So each bar in this plot is a drug. And we did a 10 cross validation and calculated the concordance index of our models. And of course, we expect that many drugs are not going to be associated with EMT, so they're going to have poor C index or concordance index. However, we found that there are many drugs that are actually showing very high concordance index, and these are like the top 10 performing models. And I'm sorry because like the resolution is bad here, but like I was gonna show you that looking at this drug, for example, our generated rule is gonna be if CDH1, SNI2, TWIST2, and HMGA2 are not expressed in the sample, then it is most probably going to be a sensitive cell line, otherwise it's going to be resistant. So it's an easy to understand rule based on that. We did the same analysis for CTRBV2, and now we are at the stage where we're trying to link between the biology of these features that we found and the mechanism of action of each one of those drugs. And finally, because like we have those large data sets, we are able like to do some external validation. So here we built models. We trained the models based on EPC 1000, and then we tested on the corresponding cell lines for each drug to find whether like we can validate these models using an external data set. And the closer you are to this line, the more consistent you are between the training and the testing, and you could say that you are validating it. So you could see that there are many drugs that are actually validated. The difference in color is because like it's true that we have 77 drugs in common between the two data sets, but it doesn't mean that we can actually validate all of them. And the reason is because of this. If we have the drug response for drug 1, the response on data 1 and data 2 to be consistent, then if we build a model on this one, it makes sense that it should predict this data set correctly because the data sets are consistent and we're talking about the same sample. However, if the response is actually inconsistent totally over the same samples, then there is no model that you can build on this one that can predict on this one. So the darker the green, the more consistent between the drug response, and you could see that those dark green are actually close to the diagonal line. So as I said, we are now focusing on some of those best performing models to look at the biology of it, and in the future, like we're gonna like not restrict the features to the EMT genes. These were as just a first test case, and we're gonna look at all the different bimodal genes and characterizing. Finally, I would like to thank Dr. Hipkins, my supervisor, and lab members for their discussions, and Dr. Lindeben and her group for their help in the biology of this project. Thank you very much.
So you mentioned, I think, in the very last slide, you're going to extend this. So if I understand correctly, you picked these EMT genes because they showed bimodality in these particular cell lines, right? This collection of cell lines. So do you have a sense of like how many genes will be bimodal if you expand this to say all 30,000, 20,000 human genes?
Yeah. So this plot actually is showing the distribution of all the genes across the 900 cell lines, and these are where these genes appear. So there are still a lot of bimodal genes that we can still explore, but we just focus on the EMT.
So where would your cutoff be, like a bimodal index greater than 1, or no anything?
That is going to be part of like the analysis that we need to explore, right?
Okay.
And at the same time, like we need to make sure that whatever we pick here is going to be translated also to the tumor samples so that we can translate it to a clinical setup.
So this plot on the left is from
cell lines, and the cancer cell lines, and this one is from TCGA samples.
So you said you're making like logic circuit things for the aging. Have you looked at any of the circuits yet? To try and interpret them, or?
So that's we are at that stage. So like here we found multiple of those circuits. So as I said, like here, the features, the rule that we got at the end is that like if those 4 genes are not expressed, then it's going to be sensitive. If they are, if it's the opposite, then like it's going to be resistant. So we're going to look at those different rules and see if they relate to the mechanism of action.
All right, no more questions. Let's thank our speaker again.
Thank you.